Hello everyone, this is Juanpablo Olguin from the University of Florida. I currently am a PhD student in the Biomedical Engineering Department, and today I will be presenting on engineered hydrogels as ECM mimetics to probe protein and carbohydrate interactions. Some background of my research is specifically on the extracellular matrix, which is a non-cellular component present within all tissues and organs. This region provides structure for cellular constituents, and it's the region where crucial biochemical and biomechanical cues are required for tissue morphogenesis, differentiation, and homeostasis. The approach I will use is to create a 3D polymer network with user-defined tunability as an ECM mimetic of extracellular protein-carbohydrate interactions. I will mix the self-assembly glycol nanofibers known as GlycNAc-Q11 and polyethylene glycol diacrylate to create a functionalized hydrogel. Q11 nanofibers have shown to undergo beta sheet formation and ultimately lead to nanofibers, and when you decorate these nanofibers with GlycNAc sugar, lectin protein WGA has a high affinity to bind to it. The design is to create these hydrogels using the pre-polymer solution of the self-assembly nanofibers, a fold initiator, as well as the polymer of polyethylene glycol diacrylate that will be exposed to UV light and therefore create these gels. First, I want to show that these gels does not change its morphology with the self-assembly nanofiber and that the self-assembly nanofibers are accessible. Through THT staining, swelling ratio, and cryo-SEM data, I showed that the presence of Q11 does not change the morphology of the hydrogel, and it is also accessible. I then want to show that the amount of protein bound into the gels can be tunable. I first showed that the presence of the glycol of the 100% GlycNAc-Q11 gel shows much more protein bound compared to blank gels, or gels without the glycosylated variant. I also showed the amount of protein bound into the hydrogels could be tuned by increasing the sugar content on the nanofibers. The more sugar, the more protein bound. As well as increasing the peptide concentration at fixed sugar concentration on the peptide could increase the protein absorption. I also showed that the amount of protein released from the hydrogels could be tuned by basically when you increase the carbohydrate concentration, there's less protein released throughout time. I then want to further modify these hydrogels for cellular interactions. To do this, I have, through an enzymatic conversion, I added a lactose component to my GlycNAc, creating N-acetylactosamine. In addition, I further modified my polymer with this fibronectin-derived peptide known as CS1 that is specifically for Jurkat cell adhesion. Throughout these 4 different composites, I showed that the composite of the CS1 peptide plus the LACNAc-Q11 gels have the most cellular adhesion. These images show that the CS1 with LACNAc-Q11 gels has the most living cells that's adhered on the gel. Furthermore, I want to use this tool to investigate cellular adhesion, migration, and viability. These are my references, and I'd like to thank my funding sources.